Hi, I'm Karsten Klingenberg, and this is my exam mini video number 5. A protein that is 5 amino acids long is formed from this DNA sequence in vivo. For this process to occur, the DNA sequence must first be transcribed into RNA. To do this, we need to identify the coding strand and which one is the template strand. To do this, we first must flip the bottom strand to allow the protein to be read from 5' to 3'. Now we can notice that both strands contain the start codon ATG or AUG once it's transcribed because all T's become U's in the mRNA sequence. The coding strand can be identified by looking 4 codons down from the start codon, and it should contain a stop codon. The top red strand can be seen to have a TGA, which would change to UGA, which correlates to a stop codon, so we know that the red top strand is our coding strand. With our coding strand, we can transcribe the mRNA sequence by just changing all the T's to U's. With the mRNA sequence, we can translate this into the amino acid sequence. The reading frame always starts with the start codon AUG, which codes for methionine. Next we have UGG, which codes for tryptophan. Then AUU, which codes for isoleucine. Next is ACU, which codes for threonine. And finally UGA, which is our stop codon. If a mutation were to happen that changed cytosine to a guanine in the coding strand, this would represent transversion and would also change in the mRNA sequence. This would result in the 4th amino acid to be changed from threonine to serine. This is an example of missense mutation because a change in one nucleotide results in a change in the amino acid sequence. This change may or may not affect the function of the protein, depending on the importance of the amino acid to the protein's function. Now, if we were to change the guanine to adenine in the 2nd amino acid, which is an example of transition, it would cause the 2nd amino acid, which is tryptophan, to be changed to a stop codon. This is an example of nonsense mutation because one of the amino acids would be changed to a stop codon and result in a shorter and most likely non-functional protein. Now looking at the 3rd amino acid in the sequence, if we were to change the thymine in the coding strand, which would be changed to a uracil in the mRNA, to a cytosine, this would be another example of transition. Changing the thymine to cytosine would cause the 3rd amino acid to stay as isoleucine in the amino acid chain and would result in no difference between them. Therefore, it is considered a silent mutation because it still codes for the same amino acid sequence. Lastly, we have transversion. We have a transversion in the stop codon, changing the adenine to cytosine. This would result in what was the stop codon to now code for cytosine in the amino acid sequence. This change would cause the amino acid sequence to not stop at the original stop codon, but would continue until another stop codon is received in the reading frame. This is an example of read-through mutation and would result in a longer non-functional protein.